Today we are going to discuss what determines the formation of spikes on the Hungarian horntail, the dragon Harry Potter faced during the Triwizard Tournament. As seen in the picture, the dragon has thorns only on certain areas of its body. In order to understand why, we're going to take a look at a gene expression pathway. Here's the proposed developmental network of spike production in dragon skin cells. It is seen that spike production is controlled by 5 different genes that create products that either activate or suppress other genes. In the pathway, an arrow represents a gene product that stimulates the production of another gene, while this symbol represents a gene product that suppresses or blocks the production of another gene. This proposed network shows the wild type or expected outcome when all of the genes are present and functioning properly. In this pathway, we can see that gene S activates gene P, which in turn suppresses gene I. This means that gene I is not able to suppress gene K, its intended function. So gene K then activates gene E, which stimulates the production of spikes. Therefore, skin cells with the wild type genotype will produce spikes and may be found on the top of the head of the dragon. Next, we will look at a pathway that lacks gene P. Since gene P is not present to block gene I can block gene K, which in turn prevents gene K from activating gene E. Without an activated gene E, spikes do not form. Therefore, skin cells with the P- genotype will not produce spikes and may be found on the belly of the dragon. Next, a pathway that lacks gene I. Without I present, gene K will not be blocked. Therefore, it can activate gene E, which in turn activates the production of spikes. Therefore, skin cells with the I- genotype will produce spikes and may be found along the spine of the dragon. Next, we will look at a pathway that lacks gene I and E. Gene E is the very last gene in the pathway, and without the activation of gene E, spikes cannot form. Without gene I, gene K would be activated to stimulate gene E, but since gene E is absent, this does not matter. Therefore, skin cells with the I- and E- genotype will not produce spikes and may be found on the legs of the dragon. Finally, we will look at a pathway that lacks genes S and K. Without gene S, gene P is not activated to block gene I, so gene I will block gene K. However, since gene K is absent from this pathway, gene E will not be stimulated and spikes will not be produced. Therefore, skin cells with the S- and K- genotype will not produce spikes and may be found on the inside wings of the dragon. Now, you may be wondering why there aren't spikes all over the Hungarian horntail's body, since its skin cells all have this basic developmental pathway. As we saw in the mutant pathways, if a mutation occurs and one of the genes does not get produced, then the pathway may not produce spikes. An example of genetic regulation would be RNA editing, like RNA splicing. In translation, when DNA is converted to messenger RNA, genetic material goes from DNA to precursor mRNA, where it gets edited further to create mature mRNA. The pre-mRNA contains introns, or non-coding regions, and exons, or coding regions. When RNA splicing occurs, the introns are removed from the pre-mRNA and the exons are joined together. There can be alternative splice sites in the mRNA during RNA splicing that remove some of the exons and result in mRNA that lacks the genetic information for certain genes. As seen in this example, the region that codes for gene P could be removed in RNA splicing when the introns are removed and result in a lack of production of gene P, therefore a lack of spikes. Due to RNA editing, certain skin cells in a specific region may not have spikes present because all of the necessary genes are not produced. Thanks for watching, and here's a list of my references.